Hello and welcome to the part of the immune system that is neither innate nor adaptive, but because it is often both. So here we go. The first thing we're going to look at is the complement system, and this is a chemical system that can be triggered by the innate or the adaptive. And so before we get into it, well, I'll talk about what the system is, but what you need to know about this system is the three ways it can be triggered and the three possible outcomes. Okay? So 3 triggers, 3 outcomes. That's what you need. So it is a system in the blood. So it protects us by protecting the bloodstream. That's the complement system. And it's a lot of proteins in the blood that are kind of unstable, and they're ready to flip into a new three-dimensional shape that will flip the next kind. They're not like prions. This is more like a signaling cascade, and they're just ready to do what they do. So they have 3 outcomes, and here are the 3 outcomes. They can cause and promote inflammation. So that's inflammation has lots of other effects on the body. So the complement system can do that. It can bring in macrophages and neutrophils. It can, opsonize pathogens. So that's a big one. And the last one is sometimes it can lyse pathogens directly. And it can be activated 3 ways. And these are just the names. So this is a fun concept map of the complement system. And what this can show you is there are a lot of different proteins, and it gets complicated, but I don't see any point at all to getting into the weeds here with which protein is which. You should know the triggers and the outcomes, okay? So let's look at the triggers of this system. One of them, called the alternative pathway, it's just that some of the complement proteins can respond to certain PAMPs. So look up, if you don't remember, what is a PAMP? That's a big deal. Stop the video, look it up if you don't remember. The alternative pathway just relies on the fact that some of those proteins respond to certain PAMPs. The MBL, or lectin-binding protein, is for things like some fungi that have mannose on their surfaces. Some pathogens, I think fungi, some other eukaryotes, I think, have mannose, a lot of mannose on their membranes. I'm not sure why, but there are proteins that, if they attach to those mannoses, they'll trigger the pathway. Mannose is a sugar, just like glucose. It's just a slightly different. And bacteria have a different relationship with it than we do, and I assume fungi do too. Well, the one called the classical system, this is the first system we knew of that triggered the complement system, and this is how it was originally discovered. When antibodies bind to something in the blood, their FC constant region changes. And lots of different cells have receptors to detect that new change. Because lots of parts of the body want to know if the antibody is actually attached to what it's supposed to attach to, or whether it's just a random antibody circulating. And so one of the things that the FC region does is it's an opsonin. If a macrophage sees that, it knows, okay, I better eat this thing as fast as I can, because whatever it is, we have a huge immune response against it. We have activated B cells and activated helper T cells. That requires a major infection. That requires tons of different dendritic cells getting the same antigens and running around showing them to all these cells. That changed FC region is an important opsonin, but it also triggers complement activation. So, just anything in the bloodstream with antibodies attached to it is going to trigger the complement system. So I'm not going to get into these two. We've talked about opsonization, and we'll look at inflammation a little bit. And you'll read about inflammation for the rest of your life because it's very, very important and not very well understood. One of the weirdest things that complement can do is some of the proteins can actually form a pore in a bacterial cell membrane. They can get to the cell membrane and form a pore. And we call this the membrane attack complex. So think about this. The whole system, the complement system, was discovered when people were doing experiments with antibodies. And they realized that if they put antibodies and a pathogen in the blood of I don't know what, somehow those pathogens died. And they're wondering, well, do antibodies kill stuff? Well, no, but antibodies trigger formation of the membrane attack complex, which does kill microorganisms. So complement has 3 triggers and 3 effects. They're kind of wild. Inflammation and fever are a big deal. They are both defense responses. Inflammation that causes a lot of different effects around the body, and it's part of healing. It's just defensive response to lots of different things. And it involves, vasodilation. It involves bringing more blood to the site of injury or the site of infection, getting immune cells there, neutrophils and macrophages. And that swelling leads to pain, so we just notice, it's painful to have inflammation. Really, the purpose of inflammation is to do all these things at the same time that will promote eventually short-term healing and protection against an injury and microorganisms that get in during an injury. So we haven't talked about natural killer cells yet, but we've talked about all these others. And notice that as more blood gets into a fluid, it's bringing the complement system with it. It's bringing antibodies with it. It's bringing T cells. It's actually bringing T cells and phagocytes. Helper T cells want to get in there so that they can talk to the macrophages. Cytotoxic T cells want to get in there and kill infected cells. A lot of this works better when fluids are invading a tissue and it's swelling. So that's what we're hoping happens with outcome, but with inflammation. But of course, we know that excessive inflammation leads to tissue damage, and chronic excessive inflammation leads to a lot of chronic illnesses and organ dysfunction. If we could control inflammation perfectly, it would be a huge improvement. We do have anti-inflammatory drugs, but if we could really finely control it, we could use it as a weapon and turn it off, and it would prevent a lot of illness. So fever is another of those kind of wild general responses, and it just increases the temperature of the body. And that accomplishes a few known effects, including faster phagocytosis. So macrophages and neutrophils and dendritic cells work better. Tissue repair is faster. I don't know why. I don't know how that helps. And some bacteria are poisoned, of course, by higher temperatures, just like we are. And so the difference is, if we go up a few degrees, our cells can handle it, but their cells slow down. They don't have as many systems for changing their behavior at different temperatures as we do. Within our allowable temperatures, we can function. They slow down pretty quickly as their temperature goes up. So that's really what fever is for. I don't have much more to say about it. So in the next video, we will look at natural killer cells, and these are wild. These are a huge part of an antiviral response. So I can't wait to tell you about them, and I'll see you in the next video.